Hello students, having studied the various aspects of metabolism, let us see the integration of metabolism during various stress conditions, namely exercise, which is normal to strenuous, starvation, immune challenge, excitement, ethanol challenge, and of course in metabolic syndrome. To introduce the topic, umpteen number of chemical reactions occur in our body which digest and assimilate the nutrients, converting them into energy and also eliminate the waste products. The course of such normal metabolic events is affected during various kinds of stressful situations. Here we are to discuss the differential metabolic events which would occur to support or challenge the stressed conditions, thus meeting energy demands by balanced or shift mechanisms of fueling. Coming to the objectives of this lesson, how the body could be set out of balance at different levels like physical, emotional, mental and pathological, thus compromising the normal metabolism, and how it could be balanced by alternative modes, and also how the adaptation to metabolic stress would differ in different various stress levels and conditions. Coming to what is physical exertion and fuels, the selective mode of fueling the body in different stress conditions highlights the essential features of metabolic integration. Fueling of the body during different forms of exercise necessarily differs. The normal level of lactate in blood is about 1 to 2 millimolar and could increase up to 22 millimoles following various strenuous exercises. However, the level could be reduced to normal levels in a time period of 6 to 8 hours, provided the body is subjected to a mild form of exercise. What happens during muscle contraction? During this, let us see, and all of you would have heard about a protein called myosin, which converts chemical energy into movement. Myosin is in turn power driven by ATP, but the levels of this energy currency in muscle is very small. Thus, obviously, the velocity of running is dependent on the rate of production of ATP from other sources. Another potential high energy compound called creatine phosphate. It's also called phosphocreatine. It promptly transfers its phosphoryl group to ADP to produce ATP. All of you know this. However, the level of creatine phosphate is minimal like that of ATP level, and thus fueling of sprinting muscle could last for only 5 to 6 seconds. During sprinting exercise for about 10 seconds, ATP levels fall to 3.7 millimolar from 5.2 millimoles, while muscle glycogen to production of more ATP, whose rate is however lower than the transfer of phosphoryl group from creatine phosphate to ADP. The level of creatine phosphate falls from 9.1 to 2.6 millimole, while that of blood lactate from 1.6 to 8.3 millimole due to aerobic glycolysis. During strenuous exercise, the active muscle releases H+ into blood, thereby lowering the pH from neutral. This mode of speed could not be continued during a 1000-meter run. This is because of many reasons. One, period of consumption of creatine phosphate lasts only for a few seconds. Number two, production of more of lactate, which causes acidosis. This scenario eventually demands alternative energy sources. Even though muscle glycogen undergoes complete oxidation to carbon dioxide and thus increases energy output, this aerobic method would be slower when compared to anaerobic glycolysis. Nevertheless, if the distance of the run is increased, the oxidative phosphorylation becomes the source of energy for 1000-meter run. Yet, the rate of ATP production is slower than the glycolytic pathway, making it comparatively slower than in a 100-meter sprint, which is shown here. Thus, the championship velocity for 100-meter event is 10.2 meter per second, while that for 1000 meter and it is 7.6 meter per second as shown here in this figure. Now let us see how an entirely varied mode of fueling is required for marathon running, which covers 42,000 meters. The body is cooperatively fueled by muscle, liver, and the organ that is adipose tissue. Though in addition to muscle glycogen stores, liver glycogen could be entrapped for energy demand. This 2 hour of exhausting event may not be sufficiently powered by the total body content of glycogen. There comes the role of adipose tissue, which provides additional ATP molecules through fatty acid oxidation. Once again, it should be learnt, and you should always remember that the ATP production by breakdown of fats of adipose tissue is slower by 10-fold than that of creatine phosphate, still lower than that of glycogen oxidation. Thus, if the ATP is supplied only by adipose tissue, the runner will complete the marathon only after 6 hours. So what happens to top runners of marathon? They utilize the same quantities of both glycogen reserves and fatty acid pools during their race in order to attain a mean velocity of 5.5 meter per second, which is just half of that for sprinting stress of 100 meter. When there is a signal of low glucose, glucagon or insulin is elevated, that could switch on mobilization of fatty acids, that is from the adipose tissue reserves that gain entry into muscle for degradation by beta oxidation. The product formed is acetyl-CoA, which is eventually converted to carbon dioxide. Further, synthesis of acetyl-CoA level results in reduction in the activity of complex of pyruvate dehydrogenase, leading to a blockade in its production from pyruvate. Thus, oxidation of fatty acid leads to diminishing of channelizing of glucose into TCA cycle and hence to oxidative phosphorylation further. What happens then? This in turn results in sparing of glucose, that is spared so that it could be available till the end of the phase of marathon. The concurrent utilization of both the fuels, namely glycogen and fatty acids, result in a comparatively greater value of mean velocity than a situation where only glycogen would be exhausted well before the utilization of glycogen. Now, let us see about starving stress. So, what happens in this period? In a period of 24 hours of lack of food intake, glycogen reserves in liver and muscle would be completely exhausted. Thus, the body during starvation has to explore for glucose equivalents to maintain normal levels of it in the absence of a direct source. The liver proceeds with gluconeogenic process by utilizing lactate and aniline from other tissues. The excessive reliability on oxidation of fats leads to increased levels of ketone bodies. It could be perceived that the muscle would not be directly involved in the starved state for the maintenance of glucose as it lacks glucose 6-phosphatase activity. Nevertheless, it could contribute by promoting the degradation of muscle protein, which in turn could be useful for liver in its gluconeogenic process. Further, the muscle also makes use of fats and ketone bodies, thus meeting the energy demands in the absence of food source. Adipose tissue functions to release fatty acids till its major reservoir is emptied. It's well demonstrated that the brain tissue relies only on glucose. However, it is for a limited period of time only. It could switch over to ketone bodies for its energy requirements, thus sparing some glucose for red cells as shown in this figure. As you see here in the table, this provides data on the exchange and consumption of fuels in a period of 24 hours. Now coming to coordinated response. What is it? The coordinated response of immune and endocrine systems on nutrient stress. The metabolic responses to nutrient stress are influenced by age, sex, nutritional intake, and of course the requirements, which are not uniform throughout the body and also across the organs. So what happens to nutrient stress exposure? The body would attempt to rebalance and stabilize the internal environment to facilitate recovery of internal metabolic responses by a series of biochemical signals. This rebalance would be achieved depending on the energy status and functional burden of the organs. As stress causes an overproduction of oxygen and nitrogen-free radicals, loss of metabolic regulatory controls such as impaired insulin secretion or hormone secretion from pituitary would lead to the manipulation of nutrient use by the various organs. Thus, stress would force the body to meet the priorities of cell as shown in this figure. The brain and the central nervous system are the organs that demand more nutrients owing to its ceaseless work. Equally demanding are the placental tissue as it demands the requirements of the growing fetus. Adipose tissues are the last in the list, that their gradual formation is based on nutrients that are excess and hence are only in the stored units. Impairment of this response would result ultimately in the reduced growth rate, reduced cost of maintenance of metabolizable energy, and also in the loss of efficient nutrient utilization for growth. So what happens to stress similar to normal conditions? There happens an excellent coordination of the endocrine type responses through the movement of hormone or cytokine in blood to distal target organ or cells through pancreas and parenchyme and autocrine systems. The output of endocrine system is affected by the prevailing immune and nutritional status. Similarly, the immune system is enhanced or repressed by nutrition and endocrine system. Both endocrine and immune system affect as well as are affected by nutrient intake. So, each of these units involved are shaped on the stress inputs received by the body as you see in this figure. The major anatomical structures that mediate stress response are hypothalamus, pituitary, adrenal gland, axis and autonomic nervous system. So following the immunological challenge, the HPA and autonomic systems are coordinated. These affect the signal transduction mode. The endocrine system is a major target for stress on immunological challenges. A variety of factors regulate hormone and cytokine secretion by glands and cells, namely from pituitary, pancreas, lymphocytes, Kupffer cells in the liver. For example, Kupffer cells released prostaglandins prior to cytokine elevation following immune compromise, and this way impact HPA by activating the receptors for prostaglandins in brain regions controlling the HPA. So during stress, the regulatory peptides and the factors are synthesized and released in brain structures like hypothalamus and pituitary gland, and of course in the thyroid and adrenal glands. These in turn affect the nutrient uptake by the organism. The cycle of interactions between the three areas finally alter the metabolism of the target organs as well as the nutrient partitioning based on the tissue requirement as shown here. Similarly, during different physiological and pathophysiological situations, in stress also, specific plasma proteases can degrade hormone binding proteins and modify the hormone binding capacity of specific hormones. Further, down at the receptor level of cytokines and hormones, there still exists modulation in the form of number of receptors, receptor ligand binding affinity and post-receptor signal transduction so as to regulate the metabolism. Adipose tissue serve as the first fuel, or you can say as the first source of energy fuel, and the skeletal muscle cells provide the amino acids and glutamine. Further, liver and muscle cells are first called in stress response so as to provide glucose substrates through hormone mediated breakdown of glycogen. For example, when cardiac muscle metabolize lactate in either exercise or as a response to immune challenge, the heart as well as kidney and intestine can serve as a sink for the elimination of these carbon sources, sparing glucose for use by other tissues. At situations of immune stress, when metabolic demand on monocytes and macrophages increases, glutamine released in muscle breakdown is the primary source of carbon for these cells for proliferation of as well as to spare glucose for other tissues. Coming to the status called excitement. Excitement is a kind of stressed state that claims glucose and energy from muscle. Epinephrine induced glycogenolysis and glycolysis in liver meet the immediate needs of energy. The increased demands of muscle are also met by fatty acid oxidation and release of fatty acids from adipose tissue. It should be understood here that the influence of epinephrine on glycolysis is different in liver as well as in muscle. The reason behind this is, while muscle needs to utilize the glucose, liver needs to produce glucose. This is effectively monitored by phosphorylation of enzymes that degrades and synthesize fructose 2,6-biphosphate, which is the key regulator. So on excitement, what happens? The epinephrine signaling should result in an elevated phosphorylation and activation of enzyme that produces fructose 2,6-biphosphate and should inactivate the enzymes which degrade it. On the other hand, in liver, epinephrine should promote gluconeogenesis, which in turn could only happen when the levels of fructose 2,6-diphosphate falls enough to promote that alternative pathway and when there is an urge to reduce levels of fructose 2,6-diphosphate. Hence, in liver, epinephrine should activate the enzyme which degrades fructose 2,6-biphosphate and should inactivate that produces it. Cyclic AMP, the secondary messenger, as all of you know, increases the phosphorylation of proteins and fructose 2,6-biphosphate, which promote glycolytic pathway while inhibiting the process called gluconeogenesis. Now, coming to alcohol and stress. Stress is induced by consumption of alcohol, of course, only in excess, and this causes changes in metabolic pathways. The metabolism of ethanol occurs in two compartments of a cell, one in cytoplasm and another in mitochondria. Accumulation of more of NADH during alcohol consumption prevents the oxidation of lactate to pyruvate, which affect the gluconeogenesis, which finally leads to acidosis. So what happens? Further, NADH inhibits the oxidation of fatty acid, thereby hindering NADH generation for ATP production via oxidative phosphorylation. The surplus NADH, however, positively influences the synthesis of fatty acids. This leads to fatty acid and accumulation called fatty liver, and this is nothing but, again, you have studied already in metabolism as accumulation of triacyl glycerols in liver. Coming to how ethanol induces microsomal ethanol oxidizing system, cytochrome P450 dependent pathway, and how it generates acetaldehyde and acetate along with oxidation of NADPH to NADP, which eventually generate free radicals. This, along with the lack of regeneration of reducing powers, lead to tissue damage. Further, in stress conditions due to ethanol consumption, regeneration of glutathione and antioxidant could not be performed, leading to intense oxidative trauma. Metabolites of ethanol also cause a massive impact. In liver, mitochondrial compartment converts acetate to acetyl-CoA, and this is done by thiokinase, which is under normal conditions that stimulates fatty acids with short chains. But prolonged period of acetyl-CoA processing by TCA cycle is obstructed by the inhibition of two important regulatory enzymes, namely isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. The obstruction obviously leads to the accumulation of acetyl-CoA that could create the following events: aggravation of previously existing lactoacidosis by formation or release of ketone bodies. Acetaldehyde pools begin to develop due to the inability of liver to process acetate. Thus, the formed acetaldehyde, owing to its covalent link with the macromolecule, namely protein, damages the functions of protein, finally causing death of the cell. Increased ethanol consumption causes liver damages such as fatty liver, hepatitis, and inflammation, and as all of you know, nothing other than liver cirrhosis, which is characterized by fibrosis and scars in the liver. Cirrhotic liver is highly functionally impaired, thus the levels of ammonia is raised in blood, which causes inability of the liver to convert ammonia to urea. Accumulated ammonia causes damage to nervous system, resulting in coma followed by death. And all of you must be now familiar with the disorder, namely diabetes mellitus. You encounter this with all lifestyle patterns. And what happens to the changes in metabolic pattern? It is a metabolic syndrome actually, where stress is caused due to overproduction of glucose in the liver, but underutilization of glucose in peripheral tissues. In type 1 diabetes, which is insulin dependent diabetes mellitus, as all of you know, there is devastation of beta cells of pancreas due to autoimmunity, thus affecting insulin secretion. Whereas in type 2 non-insulin dependent diabetes mellitus, there is an insensitivity to insulin and higher levels of glucose, which is compared to normal, is present or observed in the blood. So what are the signal changes that occur in diabetes? In type 1 diabetes, in which there is lack of insulin and the levels of glucose vary, the patient would be in a state very much similar to that of a starved state. The lack of primary signal insulin obviously leads to an impaired utilization of the crucial metabolite, which I projected as CM here. Both the processes of gluconeogenesis as well as ketogenesis would predominate in this insulin deficient state. Further, due to excessive glucagon, the levels of fructose 2,6 biphosphate increase, and as a consequence, the inhibition of glycolysis along with the initiation of gluconeogenesis would occur. This is because of the distinctive influence of fructose 2,6 biphosphate on the two enzymes, namely phosphofructokinase and fructose 1,6 biphosphatase. The two regulatory enzymes of glycolysis and gluconeogenesis are phosphofructokinase and, as you know, fructose 1,6 biphosphatase, which converts fructose 6-phosphate into fructose 1,6 biphosphate and fructose 1,6 biphosphate to fructose 6-phosphate respectively. Phosphofructokinase is regulated by fructose 2,6 biphosphate and AMP as an activator, and of course ATP as well as citrate. On the other hand, fructose 1,6 biphosphatase is regulated by citrate as an activator and AMP as well as fructose 2,6 biphosphate as an inhibitor. Glycogenolysis is also promoted in diabetes. Why? As a consequence of higher glucagon insulin ratio, thus overloading the blood with glucose. When the levels of crucial metabolite, as I said, CM, crosses the renal threshold value, it is excreted in urine. The condition described by a 2nd century physician, namely Cappadocian, as sweet melting down from body into urine. So along with CM, water is also excreted, thus making an acute diabetic to feel more of thirst, which we call as polydipsia, and hunger, which we call as polyphagia, with increased loss of water, which we term as polyuria. The stress exacerbates this condition of impaired metabolism by creating an unrestrained metabolism of both protein and lipids. Beta oxidation produces extra load of acetyl-CoA, which cannot gain entry into TCA cycle due to paucity in the levels of oxaloacetate for condensation. So it could be recalled that mammals cannot make oxaloacetate from acetyl-CoA, which is obtained only from intermediate of glycolysis, namely pyruvate. There would be more of ketone body formation. Thus, in diabetes, the body is fueled forcibly by chosen alternatives, the lipids rejecting the copious CM. When there is an excessive production of ketones, it surpasses the ability of the kidney, which would otherwise be able to eliminate in order to maintain the buffering system. Coming to the condition called obesity. All of us talk about obesity. So how the signals are affected? Let us see. The signals which are significant in stressful condition of obesity are leptin, a proteinaceous hormone secreted from adipocytes in fat, and insulin. When there is energy expenditure surpassing the food intake, like as you observe in starvation, there is a loss of mass of adipose tissue. In such states, both insulin and leptin would be diminished in their secretions, which make body to switch over to utilization of stored fuels. In the conditions of overfeed with less of energy expenditure, the signals like insulin and leptin increase, resulting in lesser utilization and storage of fuels. Coming to the status of psychological stress, this is very, very essential, isn't it? The all lifestyle pattern that we are following and the nutrient intake possibly, we need to essentially study this in this modern world. During this condition of stress, which is psychological stress, the primary signals, namely noradrenaline and adrenaline, that is catecholamines, are released from adrenal medulla as shown in this figure. The interrelationship of organs and metabolism during psychological stress that we are going to see in this figure. The brain HPA axis is triggered to release corticotrophin releasing hormone, followed by ACH by anterior pituitary gland. What do they do now? They bind to their receptors, namely beta adrenergic receptors, to produce CAMP, that is cyclic AMP, in both muscle and in adipocytes. In liver, they bind alpha type adrenergic receptors, thus producing more of diacetylglycerol and inositol triphosphate in addition to calcium. These catecholamines activate glycogenolysis in liver, thereby increasing the blood levels of glucose. Both glycogenolysis and glycolysis are promoted in the case of muscle tissue, whereas lipolysis is stimulated in adipose tissue. The metabolic scenario in muscle is to provide quick demands of energy on exposure to stress. Proteolysis also would occur in muscle, thereby supplying substrates for gluconeogenesis. The enzymes that are started are for functioning PEPCK for gluconeogenesis and glycogen synthase. Thus, what happens? The stress-induced metabolic variations, the resultant of overall cascade of events that are discussed now, thereby prioritizing adaptive response towards whatever may be the stress factor. To conclude this module, the entire module presented here describes the different modes of energy equations during different types of stress conditions such as physical and psychological. This module further describes as to how the body would cope up with the conditions by appreciating the nature of stress factors and their demands, eventually by exploring auxiliary fuels as well as metabolic interconversions.